Friday, April 21st, and we stopped on slide 12. So we're just going to continue with slide 13, and it's just more of the anatomy of the nephron. And let me just erase this a little bit. Okay, so we've already talked about the proximal convoluted tubule, which is your PCT. And then the loop of Henle is this portion here. And this is the portion that can dip down into the medulla if we're talking about a juxtamedullary nephron. And then when it comes back up, there's this little tube that's convoluted, which is why it's called a convoluted tubule. And that's called your distal convoluted tubule. And then urine that's formed collects in the collecting duct. And there are multiple collecting ducts, or multiple nephrons that drain into the same collecting duct, which then, so you have your formed urine and then it exits down at the renal papilla. Really interesting fact is that the loop of Henle, depending on where it is, you have different cell types. So right here, note cells switch from simple squamous epithelium, so very thin, easy for substances to diffuse across, and then it switches to simple cuboidal, which makes it harder for substances to diffuse through or cross over that membrane. And we will talk more about this as we continue on with this chapter. And then notice as you travel back up again, the tissue changes. One of the biggest differences between the proximal convoluted tubule and your distal convoluted tubule, so the tissues here versus here, its proximal convoluted tubule is simple cuboidal, okay, and it has microvilli on it. The distal convoluted tubule is simple cuboidal as well, but very little microvilli. So that's the major difference between the proximal convoluted tubule and the distal convoluted tubule. They're fairly similar, except for how much how much, what's the word I was looking for? Yeah, microvilli is available. Okay, polycystic kidney disease. This is a very interesting disease and unfortunate as well. What happens is that you have a healthy kidney, and over time you start forming these cysts. And these cysts, these big old bubbles, and enlarges the kidney. The cysts basically remove or kind of destroy kidney tissue. And so it gets to a point where there's very little functional kidney tissue nephrons functioning anymore. And so when this gets severe enough, the patients will says 50% of patients need to have dialysis. Huh? Is that a hand? Yes. So here's a hand. This is from a surgery where a person had both of their kidneys removed, probably had a transplant. And so, yes, they get enlarged. You can see the cysts. Right here, this thing here is a centimeter ruler. So that would be 10 centimeters across, illustrating just how big the kidneys got. So kind of like football size. And then remember, our kidneys at most are like our fist. So highly enlarged, very unhealthy, not able to function. It's the third leading cause of kidney failure. So it's a serious concern when we're talking about kidney health. Okay, so crossed out that slide, crossed out this slide, yay. Okay, so now we're going to start talking about urine production. How do we make urine? Down to the actual nephron and how that urine is formed. So, as a reminder, your nephron is the functional unit of the kidney. It's the smallest little structure that is able to produce urine. Okay? And so there are three major processes that occur in the nephron, and then we will go through each of these processes separately. So you have filtration, and filtration is happening right there at the beginning at that Bowman's capsule. When the afferent arteriole brings in blood, you're filtering out a lot of the fluid from the blood, small molecules, ions, maybe small proteins, but the large stuff stays in the blood and leaves the capsule from the afferent arteriole. So the things that are able to pass through the capillaries of the glomerulus into the Bowman's capsule, we call that the filtrate. And so that's where we first begin the filtration process of the blood. Okay, so remember what filtrate is. The second step is what we call tubular reabsorption. And very quickly, once you enter the proximal convoluted tubule, a lot of that filtrate and substances that are in it are reabsorbed back and preserved and kept. Most of the things you want to keep, like particular ions, if there's proteins that did get through, or other substances, your body will reabsorb those so that you do not lose it in the urine. Okay, and then also a lot of the water is reabsorbed too, to preserve water, for example, if you are dehydrated. The third major step in urine production is tubular secretion, and this is the last phase, and this is where you actually have substances that are near the end, like the distal convoluted tubule, that are secreted back into the tubes, back into the distal convoluted tubule. So you have substances moving back and forth as the filtrate goes through the nephron. We will have a lovely slide that illustrates this in just a moment. Okay, but tubular secretion requires energy. It's active transport using ATP. It's moving solutes or substances against their concentration gradient. So that's why you need, or against the wall, if it's the cuboidal wall, you would need a transport protein or something to help it move back into the collecting duct. So what is urine? So if we did like a calculation, urine is made up of substances that are part of your filtrate. Okay? So part of the filtrate plus anything that is secreted into the nephron as the filtrate's moving along, minus anything that's reabsorbed out of the nephron and kept in your tissues, kept in the blood. Okay? I'm not going to ask you to know this exact equation, but you do need to know those three major steps. Wow. Okay. That's a lovely loud sound. Okay. So filtration, remember this is step one. And so these are some definitions. The one that was pretty important for you to know is the glomerular filtration rate. And so this is basically how much filtrate, the stuff that has passed through the glomerular capillaries into the Bowman's capsule, so how much of that filtrate is made each minute. And that can change depending on what the juxtamedullary apparatus is doing. If you need to adjust, okay, put your phone away, please. Unless you're laughing about AMP. Yeah, I didn't think so. Okay. And then I just lost my thought. Sorry. So the amount that you were filtering out each minute in the capsule, controlled by the juxtamedullary apparatus, dependent on your fluid levels in your body, your blood pressure, and that can be adjusted. So definitely know your glomerular filtration rate. That's the one I'm most interested in you remembering as a definition. Now this is a really helpful slide because it shows you kind of those three major steps or things that occur in this formation of urine. So you have your filtration, which is number one, your tubular reabsorption, number three, and then secretion. Oh, tubular reabsorption is number two. Tubular secretion is number three. So we begin at the glomerulus, that's our knot of capillaries, where we have the fluid and dissolved solutes of the blood that filter out and then enter into your proximal convoluted tubule. So you formed your filtrate. And as the filtrate travels along the proximal convoluted tubule, you have a lot of tubular reabsorption occurring. So where a lot of the water is reabsorbed, where many ions or other substances are reabsorbed as well, you don't want to get rid of extra ions or proteins, among other substances in your urine. Your body needs a lot of that. And then as you get to so right here is the loop of Henle. So as you start traveling down the loop of Henle, this is where the cell type changes. So the thicker wall from basically here to here, it's thicker because it's simple cuboidal epithelium, but once you get about a third of the way down the loop of nephron, it turns into simple squamous epithelium. So it's thinner, and that means it's going to be a lot easier for substances to pass through that very thin wall of the nephron. And so what's commonly happening in the descending loop of Henle is a lot of water is taken back out, reabsorbed. Okay? And then the filtrate continues traveling, goes up the loop of Henle, and then as you go up, notice it changes back to that simple cuboidal epithelium, which again makes it much more difficult for substances to pass through because it is thicker. And then the last phase, tubular secretion, happens in the distal convoluted tubule. Remember, the distal convoluted tubule is shorter than the proximal convoluted tubule. And so in tubular secretion, this is where, remember, active transport occurs here. And you are actively transporting certain substances back into the nephron. Okay? And then whatever is left of the filtrate and what has been reabsorbed or what has been secreted into it is then dumped into the collecting duct and then travels down to, there's our that little dip right there is the renal papilla, and then it goes to the renal sinus. Okay, so a little bit more about the filtration membrane. We've talked about this briefly before, and this is the glomerulus. So this is that knot of capillaries within the Bowman's capsule. Remember, there's these special cells called podocytes. They wrap themselves around the capillary, and they form what we call the filtration membrane. And they leave these little slits that allow fluid and some of the smaller solutes in the plasma to diffuse out into the glomerulus to form your filtrate. So it makes it pretty permeable. And then notice, so remember, big things like blood cells don't pass through. If proteins do actually pass through this filtration membrane, most of it is reabsorbed right away. Okay? If there's a lot of protein in your urine, that is an indicator that there's something wrong. Your kidney is not functioning properly. So you don't want to have a lot of urine. You don't want to have a lot of protein in your urine. Okay. Very interesting idea or concept is the filtration pressure. And this is controlled essentially. Remember, you have your afferent arteriole that's bringing blood into the Bowman's capsule, and you have that juxtaglomerular apparatus that has smooth muscle, so it can contract or dilate and control the flow of blood and control the pressure of the blood flowing into the glomerulus. So that filtration pressure is very important, and so you have what's called a pressure gradient. So you have high pressure, higher pressure within the afferent arteriole coming into the glomerulus and lower pressure outside. So it's pushing the fluid from the blood and anything dissolved in it out of the glomerulus into the capsule. So it's kind of the same idea as when blood gets to the capillary beds. The pressure from the arterial system helps push the blood through, helps push the fluid out into the tissues to do its job. Okay, so moving on. Okay, so how do we regulate this glomerular filtration rate? So how much filtrate are we forming per minute? Okay, so first of all, we have autoregulation. So it's happening automatically, which is really quite nice. And autoregulation is able to control this filtration rate regardless of what your blood pressure is. It's able to adjust. If you have higher blood pressure or if your blood pressure has dropped, it can adjust that filtration rate or adjust the pressure of blood coming in to the glomerulus and help keep that filtration rate fairly consistent. Okay, and the way they do, or the way that is done, is with that juxtaglomerular apparatus, which is right by that afferent arteriole. And so it can constrict or relax, which helps control the pressure of blood coming in. So that is how it adjusts for changes in blood pressure, for example. So more specifically, so if blood pressure increases in your body, so systemic pressure increases, your afferent arterioles are going to constrict. They're going to constrict, and so that prevents too much blood from flowing into the Bowman's capsule, which would make your pressure really high in there. So if your blood pressure is already really high, your afferent arterioles are going to constrict to basically compensate for that pressure. If you have a decrease in pressure, you actually have your afferent arterioles dilating to have more blood coming in. Okay, so more blood flowing into the area, which will help preventing a decrease in the filtration formation. Okay, so it's trying to keep the filtration rate constant, and it adjusts that with the constriction or dilation of the afferent arteriole to help keep that filtration rate consistent regardless of what your blood pressure is doing. And so just to remember, this autoregulation is controlled by the fluid, the filtrate that's flowing past the macula densa. The macula densa is one half of the juxtaglomerular apparatus. Okay, just as a refresher reminder. Another way to regulate this glomerular filtration rate is by sympathetic stimulation. Now, just as a quick reminder, sympathetic stimulation, this is the fight or flight division of the nervous system. Okay, and so if norepinephrine is released, what it will do, okay, is going to stimulate the arteries to constrict, the afferent arterioles to constrict. If they constrict, they're the ones that's bringing blood into the glomerulus, then it causes the flow of blood into the glomerulus to decrease. So you constrict your afferent arteriole, the flow of blood into the Bowman's capsule decreases, okay, and decreases the amount of filtrate that's being formed as a result. So that's kind of an interesting thing. And remember, fight or flight, so usually something is happening in your body, or you're in some sort of situation where you're worked up, or you're exercising and you're running for your life, or something like that. So in the case of someone having shock, what happens is these afferent arterioles are going to constrict, decrease the flow of blood into that Bowman's capsule, and what can happen is it decreases the flow of blood so much that it can actually lead to kidney damage. So someone in severe shock, you want to get them out of shock as quickly as possible. We've talked about like the ischemic reaction before and shock before, but we haven't talked about its implications on the kidney itself. Now, Matt, if you are not... Are you talking about shock? You promise? Okay, don't make me come look at your computers. Okay. So please remember, this is an important thing, something I would likely ask you. So too much constriction of the afferent arteriole can lead to kidney damage. It's supposed to be a survival mechanism, but if the shock lasts too long, then you get the kidney damage, okay? Okay, tubular reabsorption, this is that step 2. So all of that before was step 1, the filtration formation. Now tubular reabsorption, and this begins in your proximal convoluted tubule. Okay, so the filtrate that was formed leaves the Bowman's capsule, enters into the proximal convoluted tubule. Okay, remembering the cells in the proximal convoluted tubule are your simple cuboidal cells. Okay, so your wall of that proximal convoluted tubule is thicker. So it's going to be harder for substances to pass across that wall. You want to remember that. Okay? So in the whole process of reabsorption, this can happen in the proximal convoluted tubule, loop of Henle, and distal convoluted tubule. So along the whole nephron. Okay? Reabsorption can happen along the entire process. Along the entire nephron. And I do have a even more detailed slide coming up that shows you exactly what's being reabsorbed or secreted in each section of the nephron. And we'll get to there in a little bit. Okay? So tubular reabsorption can happen by many different mechanisms. So right here, diffusion. Diffusion is a passive process. Facilitated diffusion also can be passive. Active transport using ATP. Symport, a good example is, remember, let's say you have a positive ion like sodium. If that is traveling across the membrane, then a negative ion like chloride, can kind of piggyback with the sodium across the membrane. So that's what symporting is two things traveling together. And then osmosis, remember, is diffusion of water across the semipermeable membrane. So in the peritubular capillaries, the blood pressure is low. So these are the capillaries around the nephron. Okay, so they are all surrounded by blood capillaries as well, not just in the glomerulus. Substances from the nephron will be transported in any of these methods out into the renal veins. So it's reabsorbed back into the bloodstream. So you will not excrete it out in your urine. Okay? Water is going to flow by osmosis. So osmosis, again, is diffusion of water. And pretty soon we're going to talk about the significance of sodium ions in our kidney. And the concentration gradient in our kidney helps significantly with the formation of urine and concentrating your urine. Okay? So after a lot of tubular reabsorption of necessary things that you need to keep in your bloodstream, like ions and proteins and other important substances, what is left, you're going to have filtrate within your nephron with a high concentration of urea, uric acid, creatinine, potassium, and other ions, kind of depending on whether you're trying to conserve water or get rid of extra water. You may have more sodium ions, for example. Okay, so here is a really, really good slide for you to study. This is that one I was talking about that kind of gives you very specific information about what's passing in or out of the nephron in each section. So over here, starting at the proximal convoluted tubule, we have our filtrate that is entering there. A lot of reabsorption back into the renal veins is going to occur there. So if we look at these arrows, so active transport is in orange, passive is in purple. So we have bicarbonate that's passing through, water that's just diffusing through, potassium. So water, bicarbonate, and potassium are just diffusing back into the bloodstream. You do spend energy pumping sodium chloride, salt, and nutrients like proteins back into the renal blood supply because you want to keep that stuff. There's a couple things that are actually secreted into the tubes, so that's the tubular secretion. And so the two things that are actually secreted into the tubes, hydrogen ions and ammonia. Don't want those, okay? And as you travel down the loop of Henle, the main thing that's happening in the descending loop, so just going down, so soon as the tissue, the epithelial cells change from simple cuboidal to simple squamous, so nice and thin, you see that the tube becomes thinner. when you have the simple squamous epithelial cells in the descending loop of Henle, and that makes it super easy for water to just diffuse across. And water, this is osmosis, this has to do with the concentration gradient. The closer you get to the inner medulla, the higher the concentration is in the tissue surrounding these nephrons. So if the concentration is getting higher and higher, the further down you go in the descending loop of Henle, then water is going to want to flow out of the tube to the area of higher concentration. So this is one way water is reabsorbed. It's because you actually have a concentration gradient set up in your kidney. Highest concentration is the inner medulla. Lowest concentration is up there in the cortex. Okay, so then as you travel and start going up the ascending loop of Henle, you have sodium chloride being diffused out. And then when you get to the portion of the ascending loop of Henle where the walls get thicker again, back to the simple cuboidal epithelium, then you're still pumping out sodium. However, this time it's active. So this down here, this is passive, where the walls are thin, and then the thick segment of the ascending loop of Henle, you're actively transporting sodium chloride out using energy. So you're getting a lot of sodium pumped back out, diffusing out and pumped back out. When you get to the distal convoluted tubule, we have some more absorption and secretion happening in that distal convoluted tubule. And so, first of all, the reabsorption of substances, the only one that doesn't take energy is water. Water is reabsorbed back into the venous system of the kidney, but then you spend energy pumping sodium chloride out and energy pumping out bicarbonate. Okay? And then you are also pumping potassium ions and hydrogen ions into the distal convoluted tubule. This all eventually will come together and make sense. Promise. So by the time that filtrate reaches the collecting duct, you have a pretty good urine formation. It's not fully concentrated yet. That final concentration of the urine happens as it travels down the collecting duct. So as it travels down the collecting duct, it's getting closer to the medulla, and the concentration in the tissue increases. So as this formed urine is traveling down the collecting duct, you're pumping out more sodium. That helps increase the concentration within the medulla. So you're using ATP to pump it out to help keep that concentration high in the medulla, to set up that concentration difference. And then down near the end of the collecting duct, you have urea that's being passively diffused into the medulla, and water that's being diffused into the medulla. At this place, it's very concentrated. So there is a lot of urea that is kept and stays within this deeper inner medulla area because it sets up a really big concentration difference with water. And so it's very helpful when you need to reabsorb extra water, for example, if your body is dehydrated. Okay, so then finally, at the very end of the collecting duct, you finally have your finished urine product, okay? So there's actually a lot of stuff that's going on in that nephron. Yeah, kidneys are amazingly complex. It's so cool how they work, too. So guys, yes, this is a good slide to look at and study. Just getting familiar with the physical changes of the nephron and what kind of impacts that has on what's passing in or out. So you really want to spend some time with this. Okay, so reabsorption, we're still talking about reabsorption, and the first part happens in our proximal convoluted tubule, which we abbreviate PCT, because who wants to write that down every time? Okay, very quick side note. My son is learning science like human anatomy, right now in his third grade class, and one of my older students, Debbie Pinto, is his student teacher. And it's so cool. And then one of his spelling words today is cardiovascular. And I just thought it was so cool because very recently we did that, and then my third grade son is spelling words like cardiovascular and kidney, and it's just really cool. So, yay, Debbie Pinto, she's doing a great job. So even in third grade, they're learning this stuff, but not at the level you guys. You guys are like you're learning a lot of good stuff. Okay, back to lecture. I just had to share that spelling word. I thought it was pretty cool. Okay, so cells, so back to real quick info, remembering that the whole nephron is made of epithelial cells, basically either simple cuboidal cells or simple squamous cells, okay? Really important to remember because all epithelial cells have the apical surface and a basal surface, a free surface and that basement membrane where the basal surface is attached to. So if I just kind of drew a very simple portion here, our little cuboidal cells, there's our basement membrane, and the surface the cell is attached to at the bottom, that's your basal surface. Here is your apical surface facing into the lumen or the hollow tube of the proximal convoluted tubule. Lateral surfaces, if we looked at a cross-section this way of the proximal convoluted tubule, I'm trying to draw a decent picture here, it would look like a hollow tube, and the wall is the simple cuboidal cells, and you can tell because we look at the nuclei to tell how many cells thick. It's one cell thick, and then you see those little microvilli sticking out into the lumen or the hollow portion of that tube. Okay? Remember, most things that are reabsorbed out of the nephron, most of it is by active transport. Some is not, like water wasn't. In most places, water was diffused across passively. There's a few others, so you want to remember that. So most are absorbed by active transport of sodium across the basal membrane. So pumping sodium ions actively across this membrane out of the nephron. Okay? And so by doing that, you end up having a lower concentration of sodium inside the cells. And the concentration of sodium and where that concentration is higher or lower plays a really important role in how we concentrate our urine. Okay. Still talking about reabsorption. So remember that the sodium was actively transported across that basement membrane. So what we'd have is sodium is being pumped out and potassium is going in. So here's our nephron. Sodium is actively being pumped out to be reabsorbed while potassium is going in to get excreted. Okay? And so we have a really large concentration gradient, and the concentration gradient that's set up in the kidney is very important, okay? Sodium levels play a very important role in this concentration gradient. So this concentration gradient is set up with sodium levels. So when they're high in the tube, that's by active transport. And concentration gradient is also helped to be set up by symport. And so remember, symport is the best example is, let's say we actively transport sodium into something. We use energy. A negative ion like chloride will just piggyback onto sodium and go with it as it's actively transported. Okay, so symport that's that kind of idea of another ion piggybacking on someone else, getting a free ride. Okay. There are other proteins that help transport molecules in or out. You call those carrier proteins. So in transporting important things like amino acids, the building blocks of our protein, glucose, our energy source, and other important solutes across the apical membrane, things that we actually want to keep, okay? Not secrete into our urine. We want to reabsorb those things. Okay. Moving to the loop of Henle. So we've done the proximal convoluted tubule. Now we have our loop of Henle, discovered by, I believe, a Dr. Henle. Usually they're named by the person who discovers them. Okay. So, talking about our little nephron, here's our loop of Henle. Woo-hoo! And so we have our cortex and our medulla, the interstitial fluid, so the fluid surrounding our nephrons. Okay, that's our interstitial fluid. Where the concentration is higher, which is closer in the deeper medulla, that's where the concentration is the highest. That's the thin segment of your descending loop of Henle and the thin segment of your ascending loop of Henle. You have a very high concentration of solutes in the fluid outside the nephron. Okay, and it's very permeable to water. So water is going to flow to the direction where the higher concentration is. So water is going to go out because there's a high concentration of solutes out in the tissue. The interstitial fluid, the tissue surrounding those nephrons, okay? It's moving by osmosis. There are a few solutes that move into the nephron. I believe sodium chloride was one of them. Then going to the thin portion of your ascending loop, it is also permeable because it's a really thin, simple squamous epithelial cell wall. So it's easy for substances to diffuse across. This is also surrounded by tissue fluid, interstitial fluid, and as you continue to travel up the ascending loop of Henle, the concentration becomes lower the higher you go. So the concentration is low up in the cortex, and the concentration is high deep in the medulla, okay? And so that kind of controls or determines which way the water is going to flow. If the concentration's high, the water is going to flow out to the high concentration. If the concentration's low or lower than it is inside the tube, then water is going to enter the tube, and that happens. Okay, so important thing to remember about the loop of Henle, we have a thin section and a thick section. The thick section is our simple cuboidal cells. They're thicker, which means that pretty much the thick segment of the ascending limb is not permeable to anything. Not permeable to water or solutes. So things are not going to be able to diffuse across, and water is not going to be able to diffuse across either by osmosis. So if anything's going to be passing through in this area, it's going to take energy. So active transport. So some solutes, sodium, potassium, and chloride, can be transported out of the tube into the interstitial fluid. So symport, we talked about that before, like sodium being pumped out by ATP and a negative ion just piggybacking out. That happens, but you're using energy to pump them out of the tube. Okay? So remember what symport is. So here we go. So by the time the filtrate has passed through the proximal convoluted tubule, down the loop of Henle, up the ascending loop of Henle, finally that filtrate reaches the distal convoluted tubule. And here's the interesting fact. The filtrate that enters that last portion of the nephron, that distal convoluted tubule, is actually more dilute than the fluid surrounding the tube. Okay? Which kind of seems counterintuitive, but there's a reason why. So, now we're going to talk about why that. So remember, by the time we're in our distal convoluted tubule, so here, let's say this is our distal convoluted tubule. I know it's so beautiful. I'll try to make it look like a thicker tube. So it's saying the fluid inside is lower in concentration than the fluid outside. Okay? Oh, I can't write that. There we go. So this will have implications about which direction ions are going to be moving or which direction water is going to be traveling. Okay, because you have a low concentration in the tube and a higher concentration outside. And this is actually in the cortex portion of the kidney. So what happens in the distal convoluted tubule to kind of help rectify the situation is that, again, we have symports of sodium and chloride across the membrane. And so that is traveling in. Okay, so you're pumping sodium in, chloride piggybacks on that into the distal convoluted tubule. Okay, that helps to increase the concentration inside the tube. And so the concentration gradient of sodium depends on how much can actually be transported into the tube. How much can be transported across that basement membrane, that little membrane that forms the outer wall of the distal convoluted tubule. So, let's see. So because we have active transport of sodium into the cell, that leads to the concentration difference changing. The fluid enters your collecting ducts, and remember that's moving deeper into the medulla, and in the collecting duct, going down to the medulla. At first, the very beginning of the collecting duct, it's the filtrate, the urine that's almost completely formed, is still really dilute. So as it moves down the collecting duct, getting closer to the medulla where the concentration is higher, then we start having more water moving out. And that helps to concentrate your urine. You also have other substances like urea or sodium chloride that also is being transported back out to help keep that concentration high in the medulla. Okay, let's see. Yes, so here it says water moves by osmosis. That's what we're talking about right here, because in the medulla it's very concentrated, so water is going to flow right across the membrane to the higher area of concentration. And so by pulling water out of the collecting duct, you're making your urine more concentrated, okay? And controlling how concentrated or how dilute your urine is under the control of hormones, like the renin-angiotensin-aldosterone mechanism is one of them. Oh, what's the other one off the top? Oh, antidiuretic hormone is another one that will affect that. So those are two hormone mechanisms that can control this. Okay, so changing in the concentration. Oh, I should not have touched that. Is it going to come back up? Okay, there it is. All right. So, as you are traveling down that collecting duct towards the center of the kidney, that higher concentration in the medulla, the filtrate decreases because you're losing water. Water is leaving. So it makes the filtrate that's in the collecting duct get more concentrated. So you're concentrating the waste products and you're pulling back and saving all the things, useful things like the water or urea to help keep the concentration high in the medulla, and the sodium chloride to keep the concentration high in the medulla. Okay, so things that become concentrated in the filtrate and are going to be excreted, so urate ions, creatinine, sulfates, phosphates, and nitrates. Some of them are reabsorbed in the medulla to keep the concentration high, but most get sent out. Most are filtrated out in the urine because they're toxic to the body. And I don't know if you remembered in lab, we once talked about our blood has a degree of dirtiness to it because not all of your blood can pass through the kidney at each pass in the systemic circuit. So there's a certain level of waste product in your blood. And that's very normal. If it gets too high, then it does become toxic. And that's an indicator that your kidneys aren't functioning properly. If it's bad enough and their kidneys aren't functioning, then a person may need to go on to dialysis and have a machine clean their blood for them. That's really not an option most people really want to do. Okay, Tubular secretion, this is that final stage. We had filtrate formation, tubular reabsorption, and now it's this tubular secretion. This is number 3, oopsie there, on our list. And so in secretion, this is where we're secreting substances from the interstitial fluid of the kidney back into the nephron, okay? So if this is part of our tube, secretion is secreting substances back in from the interstitial fluids. Okay, this can be both passive or active. And so one of the things that diffuses into the nephron is going to be ammonia. You do not want ammonia building up in your blood, and it's very quickly converted to urea in your liver, if you remember that. And then the urea travels in your bloodstream until it gets to the kidney, where most of the rest of that is secreted out as urine. Some of it's kept to keep that concentration level high in the medulla. Some things like hydrogen ions, potassium, penicillin. So if you've taken antibiotics like penicillin, you can tell someone's taking it if you test their urine. It's secreted out in the urine, which is really kind of interesting, and it's an active process. Okay, so urine concentration. So when you drink lots of water, what is going to happen to your urine? It gets clear. So what does that mean about the concentration of your urine? So it becomes more dilute. Excellent. You guys are so good. And so one of the things that your body has to take care of is getting rid of that excess water without losing really important electrolytes like sodium and chloride and calcium. Those are just a couple examples. We need to keep those, else it's going to throw off our whole osmotic balance in our blood. So we have to get rid of the water and keep the important electrolytes. And so kidneys make dilute urine, and then they keep those electrolytes. Also, you will have some serious problems. And then on the other hand, if you're not drinking enough, your kidneys are going to reabsorb as much water as possible back into the interstitial fluid, back into the venous system of the kidney. And so therefore, it's going to make really concentrated urine so that you can conserve as much water as possible, okay, but still able to get rid of waste. Guys, we're doing so good. Okay, we'll finish this slide and we'll be done for today. So I've mentioned this multiple times. The kidney's ability to actually concentrate urine really good depends on the concentration gradient in the kidney itself. Remember, it's more concentrated in the medulla and very low concentration out in the cortex. So as those collecting ducts travel down, it's higher and higher concentration, causing water to diffuse out by osmosis and other substances also. So it's the concentration difference within the kidney itself that helps us be able to concentrate our urine in the first place. So really important setup, okay? All right, so we'll start on slide 33 on Monday. We will finish this Monday, okay? There's no homework this weekend. Remember, you have an exam on Wednesday, and there are office hours on Sunday, and if there's more, I will make sure to let the whole class know in an email. Thank you.